So I build gadgets, so I'm not a scientist. I know that sounds horrible to say, and I say this with love. It's just that I'm an engineer in the sense that, what do I mean by that? People get pissed off sometimes when I say that. I mean, my life is not about hypothesis, validation or investigation. I mean, I do that sometimes on the side, it comes out, but I just like building stuff. That's the truth. I like building gadgets. And I won't go through this, but I like building all sorts of gadgets. I think the things that have had the most impact, certainly in the last decade, have been in neural engineering, all sorts of things that have to do with building different electrode configurations and all sorts of grids and other crazier stuff that I'll get to near the end. But I also like, a variety of medical implant things that we've looked at in the lab. This is, for example, a type of technology that you can put into a bone to track how the fracture is healing. Then the crazy stuff Mike talked about a while ago that now a former postdoc who's a professor does, which is implanting electronics, essentially radio-operated neurostimulators and neuromuscular stimulators to fly living insects around like little airplanes. That's basically the idea. So we were the first to show that, and that's pretty nutty. And then other things, Daniel Cohen, for example, who's now at Princeton, really in my lab worked on all this, sort of looking at cells, almost as a shepherd, sort of herding them around and giving them cues to have them act as sort of advanced functional material. So I have a shiny object syndrome problem, and So okay, with that introduction to who I am, and sort of by inference who I'm not, let me launch into a story. And so from here on, I'll just really introduce the NeuroDust story. I'm going to give you a sense of how I see where the world is writ large, and this includes even non-academic exercises, and leave it as an exercise for you to think about it a little bit. We'll come back if people have questions in more depth about some of the commercial stuff. And then what I want to do is, and I recognize that probably, my guess is for a full third of you, the next part of these slides, which are a little tutorial to get everybody on the same page, is going to be super boring. I mean, if you're an electrophysiologist or a working neuroscientist, some of this stuff is going to be like yawn, yawn, yawn for like 9 slides. But I found that if I don't do this in general talks, I can't get everybody up to the same point, and then people then just appreciate it. I go, I guess that's cool because he says it's cool, and then sort of phase off. So I bear with me if you are technical, and I apologize. So we're in the middle of a big gold rush right now in neuroengineering, for good and for ill. I think these things have perils. Looking at it mostly from a commercial lens, not as a neuroscientist, and I'm not encouraging this, I'm just showing you an alternate viewpoint that usually doesn't get expressed in talks like this. What's happening is that the technology and the knowledge base in the sciences have gotten far enough that we can start to envision not the traditional open loop, single therapy target, very signature device plus application play that, for example, classic Medtronic is has been doing, but really pushing the envelope to try to both move into closed loop applications, which have notoriously been hard in industry, peripheral nervous system applications rather than central nervous system, which have an enormous market that I'll touch on at the end. These are really big deals, actually, that often in some classical neuroscience contexts, they don't get a lot of attention because it's not the brain, and there's an enormous amount there. And eventually, the more aspirational sort of research in a company approaches that really aim to some form of high-density interface. And this is very contentious and very fraught as a claim. And I'm happy to, again, in questions, I'll give you a taste of it. But this is really, right now, there's quite a lot of activity, and that can be both good and bad. And NeuroDust is sort of riding along in that wave. I certainly don't want to say that it informs it causally, nor does it benefit, sort of parasitically. It's like all things, sort of riding along. You're kind of surfing along on a wave that's kind of self-propagating because of the work of, thousands of people. But it's a very exciting time right now to be in neural engineering. So here's what I want to do, and actually this overview misses some stuff. First of all, my partner in crime has always been, for the better part of the last 10, 12 years, is Zeke Carmena, who's a brain-machine interface pioneer. And he and I have long ago developed a very, very close personal friendship. Our families are friends. And out of this came just a lot of creative work. And so I always acknowledge him because I'm indebted to sort of, the types of stuff he does and his vision. I'm going to start by giving you an introduction to what NeuroDust is so you appreciate it. When I tell you what it is, for those of you that don't know and just heard about it, you're going to be like, oh, that's it? Like, this we gave his due to talk. The two people that did all the work that I get to come to nice talks are DJ So and Ryan Neely. DJ So is today one of the founders of Neuralink, and Ryan Neely is was employee number 1 at IOTA Biosciences. So these are really high-powered people, and very grateful to stand on their shoulders. I'll then tell you about a modification to that is called stim dust in the academic literature. That was pushed forward by a brilliant assistant professor, circuit designer named Ricky Muller. She really led the effort to take what the original neural dust concept was, and I'll show you what it is, but modified in a way that's very useful in applications for stimulation. And again, a whole slew of extremely talented people. And then I'm going to dive into the crazier stuff. So really, this is all in the past in a sense, although, certainly Ricky continues to do amazing stuff here. Once you get there, you stop and you ask, what's the hard problem? What hard problems are left, right? What's the next shiny object? And I'll tell you about what that is. And then I want to show you something in 3 or 4 slides, which is super cool, just because most people would never have thought about it, and they're the brain product of another postdoc who's now at Carnegie Mellon. And then, because I'm ambitious, I added 3 slides on work that is 1 week old. It was just presented last week at the premier Circuits and System Conference, which I think you all will find fun. So we'll see. I'll try not to talk like the Micro Machines commercial. Well, no, I'm dating myself. So that's from the late 80s. OK. Half of you don't know what I'm talking about. All right. So this is an image of the brain I stole from Google with that attribution. When we started this journey, the motivation was, hey, can we build smaller and smaller? Can we build extremely small implants for cortical applications, in particular brain-machine interfaces? A very narrow. I'm sort of narrowing it down. So cortex. That's how it started. It's not going to go there, but cortex. Brain-machine interfaces, which means I'm not trying to do neural pixel level, 100,000 channel stuff. I'm trying to ask, what can I do that might live in a brain for a long time? And electrophysiology. So we started from a desire to extremely miniaturize essentially what is a standard EFIS voltage, extracellular voltage recording system. So here's another image I stole from Google at attribution. It's a little drawing of a neuron. Now, why does this matter? Seems pretty silly to start here. It matters because of this. So I think everybody here know that among the many, many great things that neurons do, and I don't want to overemphasize this. We were just having a conversation about how great synapses are. We all know that in part, the way information propagates is having this membrane action potential, blah, blah, blah. And we all, I think, know that shows up phenomenologically as a difference in chemical potential between the inside and the outside of the membrane as it propagates. And because Faraday's constant is a wonderful thing, that chemical potential looks like an electrical potential if I'm an electrical engineer and I want to try to sample electrical voltages. And if I were to do that, we all know a couple of enormous prizes were awarded long ago that there's tens of millivolts, close to 100 millivolts of difference, of change in that voltage reading that reflects the chemical potential if I were to be able to perfectly sample it. But of course, we can't. So how do you try to sample in a neuron, out of a neuron this? Again, 3/4 of you know this very well. Well, I would like to have one electrode, right, one piece of metal in contact with the inside of the cell and another one with contact in the outside. And in that way, I can measure that plot I just showed you. It's a very big number. Millivolts are big units. And so that's great. That's fantastic, except many of you know that's incredibly hard, right? Getting that reading is extremely difficult. And it is, in general, not applicable to the clinical realm. In other words, it's restricted to beautiful laboratory hands, amazing work, great science. You're not going to patch clamp someone in the clinic, and certainly not chronically. So what long ago people realized was that, well, what about this? So let's put the two little pieces of metal outside. Now, naively, you might say, that's crazy. You're not going to measure anything. Well, you will, because those two things are not at the same point. They're slightly offset from each other. And so as the cell goes about its business shuttling ions, those little currents are going to create echoes of that big chemical potential difference in the external milieu, and they literally are tiny echoes of the actual spike. They're not the spike, they're tiny echoes, and they're much smaller. And so what people do, they don't do it this way, because that would be doing it hard for nothing. What you usually do in conventional preparations is one electrode is very close, and something else is going to be as far as possible so that you get the biggest potential difference. You maximize as much as you can. And often that other thing is bolted to the skull or some large counter electrode, so on and so forth. And why am I telling you this? It'll become relevant to where the limits of neural dust are. Literally just out of these cartoons, you'll be able to understand it. So this is today's, everything you see that are called probes, recording probes, is some sophisticated version of, I need these two things, and then I need to have something that measures that V measure, extracellular, right? The problem, of course, is that that neuron isn't alone. It's sitting in around a whole bunch of other neurons. They're all firing. They're all causing echoes. They're at varying distances. People like to call that noise. It isn't noise at all. It's just cacophony, right? It's like the cocktail party problem, but for voltage sampling. So now you have this sort of mess, and those things have structure to them. This is a rat barrel cortex picture that's very famous. I don't know why the attribution didn't come up for that, because I did have attribution for this. This is a wonderful paper. But these things all have structure, and that structure will also create structure in the electrical signal. I think, again, this is mostly for the students in the room. The fact that all these neurons are arranged, even if they're not as beautiful as mouse barrel cortex, obviously they're not in mammalian cortex, but nevertheless, that the idea applies, right? That you have structure, and that structure will give rise to structure in the signal. And so from that tutorial, you can appreciate all the different things that people, this is from Schwartz's paper from 2007. Again, what the heck's going on with all my attribution? That's really horrible. And even the next one isn't there. Something weird happened. I apologize. But this is from a Schwartz paper from 2007. And I'll go very quickly. Again, this is for students. What's EEG? EEG is the idea that, well, let me go backwards. What's single unit recording? It's the thing I just showed you. You stick a wire in, and you get as close as you can, and you get as close to one neuron or a small subset of neurons. But of course, all those neurons are doing something together, and that was long ignored. A long time ago, people thought that was just sort of background garbage that had no content, but in fact, that's completely wrong. That's a field potential. It's the ensemble result of all these things shuttling ions around. That electrical signal has information and can be used for drive brain-machine interfaces. You can go one level beyond that and say, OK, I'm not going to do this. This is nuts. I'm going to put an electrode outside of the cortex and lay it on top and see what happens when these different parts of the cortex fire. What does that look like? That has utility. That's called electrocorticography. It was started in brain surgery, super useful, and has since migrated to enormous stuff. Look up the work of Eddie Chang and others, just really, really beautiful work. And then finally, EEG is like, I don't want to drill a hole in anybody's skull. I'm going to put those electrodes outside the head. Let's see what I see. And what you see is akin to, the Cal Stadium having a football game, and I'm 3 blocks away, and all I can hear is when they're all singing the same song. I can kind of tell that's happening. I certainly can't go in and listen to anybody's conversations. So they all have utility. These things have all been around, and over the last decade, there's been a lot of desire to accelerate this stuff. And so I'm going to skip through this because I want to get to the meat of it. But for those of you that don't know, this is an example of the surface electrodes I was talking about, electrocorticography. This launched, I launched a company off of this that did pretty well, which was just sort of wireless electrocorticography with Ricky Muller. But this is the kind of thing that was sort of the bread and butter of neural interfaces. And if you wanted to go in and do that extracellular recording, you had a variety of torture devices that are very popular. The top right one is the canonical Utah array. Some of you, I'm sure, have one here in your labs, not on you. The bottom right one is a microwire array, which is much more common. These are there's more. There's an infinite variety, sort of a torture chamber museum of things. But what all these things have is little electrodes or wires that are going to be pushed. You're going to push these things into the cortex. You're going to damage the hell out of everything as you go along. The more, you all probably know this, the more innervated a part of the brain is, the more vascularized it is, and so pop, 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 pop as you go in. And you're going to then record. And whether, if this was a super specialized talk, we could have a really fun fight about whether these side probes are any good versus microwires or whatever. But it's all that same idea. And today there's, the a number of other probes. I'm going to skip all of this. But the idea, so this is where the field was when NeuroDust was conceived, in that you had these different array things I was talking about, these sort of different ways of recording that were very, very valuable. So I want to show this lest you think that these things are all neuroscience tools. There were, the BrainGate trials implanted these types of objects into people, into patients, and demonstrated that you could record from the cortex and use those recordings, in training loops, right? You could use those recordings to train, to give individuals, a certain prosthetic functionality, right? And I use all those terms. I'm being cautious because the details of each of these studies are actually very important, and I find that people sometimes kind of resort to hyperbole. These are amazing, and I would encourage all of you to, if you haven't, to watch the YouTube videos of this, because it's important to connect to the people. You see them do this, and it's really meaningful. I mean, you see what a difference it makes to suddenly go from zero mobility to having put an enormous amount of effort, because the training that they go through to do this is astounding, and over months be able then to will say this hand to move a chocolate bar or a cup of water or whatever. And this is the field that Jose Carmena, was largely, really comes out of and in many ways pioneered. So this was the state of the art. As a gadget builder, I was looking at this, right? Look at these systems. These are amplifier boxes that are doing that, connected to these arrays, right? And I wasn't the only one, obviously, but that was the state of the art. And so, let me see how I want to do this. Okay, so one of the things let's pause and you say, okay, great, thanks for that, Michel. What's the point? Well, the point is, if you really believe that you can do better than that, or that there's an opportunity to really do some important stuff here, you have to take a pause and say, what are you trying to build? What do you want? And if we're focusing on clinical utility, okay, and only that, we're not talking about neuroscience, fundamental research at the moment. You want to have, you of course want to be able to see what you want. That's pretty trivial. And you want to have these things last. So I'll tell you right now that in the subtext of all of my talks is the following assertion, and I know no one will be able to argue. Today, there isn't a single implantable neural technology that will last an appreciable fraction of a healthy adult's lifetime, period. Think about that, right? So of course, it doesn't mean those things don't make a difference. They do make a difference in people's lives. But the idea that today humans know how to build a neural interface that I can put into a healthy 20-year-old and expect when they're 80 that thing will work is pretty close to zero. That's profound if you think about it. And the issues are very complex. So you damage the tissue when you put things in. That was focused on for a very long time in these fields. The vascular damage, the immune response, the local versus systemic, the glial scarring, all of the responses you get. But it's just as important to observe that the material challenge is actually rather enormous, right? So let me put it, I always flip it around and I use this analogy and everybody goes, oh, wow, of course. Okay, let's say I told you, how many people here are electrical engineers? Anybody? Did anybody sneak in? Okay, so one person snuck in, the rest are neuroscientists. This only makes sense to you. All right, so no. So let's say you're a circuit designer or a chip designer. I tell you, make me a chip that does whatever, adds 3 numbers. Tell me when you're done. Okay, so you give me this amazing chip that you paid, $500,000 of upfront capital or something. And then I tell you, throw it in the ocean, and I want it to work for 20 years. And you're going to look at me like, what are you, out of your damn mind? Like, what, I just spent, what? This is what you're asking of this, right? You're asking to put these very sophisticated silicon systems into the brain in essentially what are aggressive saltwater environments for decades. And so the material challenges are very strong. And I'm not going to talk about that today. I have a whole part of my group that's published on this stuff. Then you want to minimize infection. All of those wires, no matter how you treat the patient, no matter how nicely you do the preparation, are always routes to infection. This is sort of an open secret in anybody that does this. And you want to allow, awake behaving, this is sort of the paper, the journal paper way of saying it, but you want to be able to let people live their lives, right? I mean, you can't have this just dragging around this thing, right? And so the field really, about a decade ago, began to really look at this, very deeply. And the first pass of these things was, well, take all these things and make them wireless, right? Okay, thanks for the giant box. I don't need that giant box. I'm a circuit designer. I'm going to make a chip that has all these amplifiers. And by the way, I'm going to make a radio, and I'm going to make an electromagnetic coil, and blah, blah, blah. And I was part of that. And that works okay. Oh, This is here for the Neuralink fans. Let me come back to that. That slide is literally just humble bragging, so I'll come back. I'll brag later, because I want to get through this story a little further. So you say, okay, I get it. That stuff is crazy. Let me miniaturize it. And you do. You try to build a little radio, a little coil, a little antenna. You have all sorts of really cool ideas about this. And what you find is that works okay if you restrict yourselves to objects about the size of a USB stick or a little postage stamp, and they're not very deep in the body. Now, why is that, right? And I think intuitively you know this. Every single one of you look at one of these little devices, right? Nothing's really much smaller than that. And why is that? It's actually pretty simple. There's two reasons, and they're both very fundamental. And this is the beginning of the neural dust story. Like I said, when I get to the point in a minute, the punchline is about 2.5 minutes out, and you guys are going to be like, oh, really? OK, number one, we're all bags of salt water. So electromagnetic fields don't like passing through you. They like getting absorbed by the salt water, OK? Right? So you have a problem. The absorption is enormous. And so you're trying to use electromagnetics to power and communicate with something that you would like to have untethered inside the body, right? Remember, I'm sort of doing this by implication. I'm not being explicit. What I'm going to propose in a second is that there's a better way to make extremely small implants, and that's going to solve some of these problems. The other problem, and this is less intuitive, but I think you'll appreciate it. The other problem is that light is very fast. OK, what the hell does that mean? All right. Anything that's a radio or an electromagnetic coil that communicates, even if it sounds really cool, like 60 gigahertz radio, whatever. All that stuff is photons, and that means that that information is traveling at the speed of light. So what happens why is that a problem? It sounds kind of the opposite, right? It would be amazing. The faster something travels in a medium, the bigger the wavelength, right? Really fast, so you get a big wavelength. The problem with that is that when you're trying to capture energy, you're doing it through an aperture. It doesn't matter if you're talking about a telescope or a radio or pressure waves or ping pong balls. Whatever it is that is your collector, which colloquially you would call an antenna or an optical aperture, is an aperture. And that aperture has to be in a reasonable fraction of that wavelength. Otherwise, you're really bad at capturing the energy, right? It's kind of common sense. So light is really fast. And so what happens when you go and you do a thought experiment, so here's the thought experiment. I love that I can make little radio-operated, little radio implantable objects. They're going to be great for neuroscience or neuroengineering. So now I do this thought experiment. Let's make a little antenna or coil and connect it to a little circuit. That's going to be RX. That's the receiver. And I'm going to put that in the body. This is a thought experiment. I'm going to put that in the body. And outside, I'm going to put whatever coil I want, whatever the biggest, most juicy thing I can do that can talk to that. I don't care about that. I don't care how big the thing is for this thought experiment outside the body. And then I'm going to ask, how well can I get power to that little coil that I'm going to implant? Or conversely, how efficient a communication link, those two things are related. How efficient a communication link can I build with that implant as I make that implant smaller and smaller and smaller? So what you find, and this is, you can build, the physics of this is very well understood, so you can build very robust models for this. And what you find is, at a centimeter, you're fine. It's all good, just postage stamp. And you have to make assumptions, right? Here, I'm assuming it's 2 millimeters transmission distance, but you could make this much farther, whatever. I mean, the point is the shape of the curve. It doesn't really matter, what specifics you care about. The problem is that as soon as I start getting into the millimeter range or lower, right, even if for each of these sizes I pick the ideal resonant frequency for that coil, so I optimize it, I find that I fall off a cliff. That's a log scale, either in absolute power or in efficiency. I think I have plots in this talk for both. But the point is that it's a really bad idea, and you can play tricks. There are very smart people that have found ways to do things around here, but it's sort of, in my opinion, sort of squeezing water from rocks. OK, so why is that? Light is fast. You're a bag of salt water. OK, so that's what you're going to remember. And the total amount of power, if you do these calculations, you can get is like absurdly small. It's very hard to do anything useful with it, blah, blah, blah. All right, so what's the answer? So the answer is pretty simple. It's to switch the whole paradigm around and just use ultrasound. And so here's how this works. This is the only slide you have to pay attention to. Then you can go back to checking your email for those of you that are here by compulsion. If you're here by compulsion, this is the only thing you have to pay attention to. Then you can go back to doing whatever it is you're doing. So it turns out that there's a class of crystal that's been known for a long time to have a really simple property, wonderful property, which is that if you vibrate it mechanically and you put electrodes on two sides of that crystal, right, and you make the crystal vibrate, some of the mechanical energy will be transduced into electrical energy. Okay, it's called a piezo crystal. By the way, as we'll see in a second, that is symmetric. So if I apply electrical energy, it'll be transduced into mechanical energy. So all I do is I take a piezo crystal, I put it in an implant, and I hit it with ultrasound. And what will happen is the ultrasound will power up that crystal, and the crystal can power an implant. And you might say, OK, that's super simple. Like, is that it? And like, no, close, but not quite. Wait, the slide's not over. Then you can leave. No, I'm kidding. So 30 more seconds, man. All right. So what happens, though, there's one other trick, and this is actually the more entertaining trick. The other trick is that if that thing the crystal's connected to that's providing power to it, let's call it a chip, doesn't matter. It's drawn as a variable resistor there because it doesn't matter, really. It depends on what you want it to do. If that thing can change its own electrical impedance as seen by the crystal, right? What happens is then how the crystal vibrates, in a sense, will change as well. And because it's a symmetric transform between the electrical domain and the mechanical domain, what the crystal looks like to the rest of the universe also changes. And what that means, and this is the important part, is that the amount of reflected energy is changed every time the chip modulates its impedance. So what you can do, and this is the fun part, you hit it with ultrasound. Some of that ultrasound is used to power the chip, but some of it's going to reflect back by pure reflection, right? It's a sound wave. Except when it reflects back, you quickly modulate the impedance, and the amount of reflection changes, and you can encode digital information in that. And so now the thing, without any batteries, without anything, can communicate back information at a pretty damn good rate. And you can make that analog. I'll show you the analog version. You can make it digital. I'll show you Ricky's version, which is a digital version. That's the point. That's literally the whole point of neural dust. And why it's important is because if you work out the physics of this, these objects can be absurdly small because of the physics of ultrasound. That's it, because sound loves going through your soft tissues, doesn't care. It cares about bone and reflections. We'll talk about that later. Sound is slow. So the wavelength of 10 megahertz ultrasound is on order of a couple hundred microns. It's perfect. Right? You can build very, very efficient ultrasonic devices at millimeter and sub-millimeter scale. And again, you can do tricks like this to not just power, but have it talk back to you. So now you can already imagine, right? Like once I stop here, if I took a breath, everybody thought about it. They can go, oh, OK, I get it. You could record with this. You could stimulate with this. You could do all sorts of other stuff with this. That's the whole point of dust. Does that make sense? Yeah? All right. Yeah? Nobody cares? OK. So now I have to validate this a little bit, and the only way, I mean, I can say anything in a slide. I think I'm pretty good BSer, so you should go look at the papers. I'll give you some snapshots, but the reality is that if you do the same sort of link calculation with all things being equal and so on for ultrasound, it's just much, much better, and there's no equivalent cliff. I mean, you can really, at least in theory, go down to absurdly small sizes and still have useful links and useful power. And this is where when people ran away with this idea, is because this is true. I mean, the physics of it supports this. And people are like, oh my God, nanometers and downloading brains, and so it just quickly became crazy, right? And so what I want to show you now is, after kind of selling you that this is, and I'll show you the data and all the things in real life in a second, but conceptually, even though this is a good idea, I would like to show you now where it breaks, because that way I can also, sort of, oh no, did I not have this? These slides don't have where it breaks. OK. Let me do it verbally. I can do it verbally. I'm sorry. This is a screw up of mine. This talk didn't have that slide. The next stuff is all the data. Let me explain to you real quick how it breaks, because this is really important. So the problem is that, and this is why I did the little tutorial at the beginning. Here's the problem. If I'm doing this to, say, record neural recording, as I shrink the size of this object, I don't have, one electrode at the recording point and some other electrode really, really far away, like I showed you before, like every eFIS setup. Those two electrodes that record are stuck on whatever little object I'm making. And so at some point, if I'm telling you that I have a 50 micron object with two electrodes near each other, the farthest those things can be away from each other in a cube is the diagonal, right? And that's a very small number. And so more conceptually, and you can look at an archive paper, you can actually plot, you can work out the numbers, the math of, as I shrink these two electrodes, as I shrink the distance between these two electrodes on a given object, how does the magnitude of the measured extracellular potential scale, and it scales non-linearly. It craps out really badly, right? At the same time, because the total volume is smaller and smaller and smaller, the total amount of power I can get to it is shrinking too, right? It doesn't matter that it's efficient. I'm making it smaller and smaller and smaller. Even if I keep cranking the frequency up to match that size, the total power is a function of the volume, and so it's going to shrink. And the amount of, because I have two things working against each other. One is I make this thing smaller and smaller and smaller. The signals are getting tinier that'll show up on the electrodes, but also I'm getting less power. At some point, those two curves meet, they intersect, and beyond that point, it won't work. And that's about 50 microns. So that's the bottom line. There's no nano anything, right? Neural dust below 50 microns. There's some tricks you can play, and we've talked about it, and I think in some future they'd work. But the reality is that's what's realistic. And what is absolutely trivial to do, and this is what's so disruptive in the peripheral nervous system field, is millimeter and submillimeter objects, which are unheard of today. That's really easy to do, and I'll show you that now. Okay, so it started in the cortex, but we decided, okay, let's see what we can build. And so what we did, I'm going to skip through a little bit of details, but anybody that feels dissatisfied can stop me afterward in the questions. I asked a question that actually ticked off a lot of circuit designers, frankly, which is instead of putting a very sophisticated integrated circuit, what it was the sort of dumbest thing you could possibly connect to one of these crystals, and that's a single transistor. It's absurd. And what we did is we connected that single transistor. That's that symbol there. You don't have to worry about it if you know what I'm talking about. It's just a single transistor. And we're going to connect that to the extracellular fluid, whatever we're trying to record. And we're going to connect another terminal to some other part of the circuit. If you're not an electrical engineer, it doesn't matter. Basically, it's a crystal. It's one damn transistor, no processing, nothing fancy. And what we're going to do is build this thing. And we did. We built this thing. It's here. It's super. This is a 1 millimeter bar. So this is the original cube of the very first one we built. This is our one transistor. On the bottom are these two little recording pads. And we built these little objects so that initially, when we first implanted them into an animal, you would have wired leads that connected to these recording pads, so we could take ground truth recordings, just like if you were doing eFIS, before and during we were doing the neural dusty part of it that I'll show you, so we could get a really literally direct side-by-side comparison of how good this was in terms of reporting back neural data. Another thing I'll say, which I don't have time to talk about right now, is that, although you'll get a taste of it in 10 minutes, the systems that sit on the outside of this, that do all the ultrasound signal injection and calculation of the reflections and stuff, those are actually rather sophisticated. And I'm going to give you a taste of how sophisticated they are in 10 minutes. But there's a whole enormous endeavor in building these things that sit out the outside and talk to the dust. And so in the very first experiment that worked, we took one of these little motes we made. We put it on the sciatic nerve of a rat. We also put it on the muscle, but I'll skip all that. And what we would do is we would stimulate, electrically stimulate, distally, we'd electrically stimulate the nerve just to get different magnitudes of signals, compound action potentials propagating through the nerve, at specific times. And then we use this dust to ask, hey, does this whole crazy thing that we're talking about actually work? And it does work really well. And what I want to show you is for that early version, that sort of 2016 version, I want to show you how it works, because I think walking you through this will give you an appreciation of the fundamentals. Don't be intimidated by this. I promise you it's not hard. A lot of this stuff, this is straight out of the paper, but look at column 3. So what this system is doing is every 100 microseconds, you see that bar below the blue curve, it sends 6 pulses of ultrasound at the crystal's frequency. So it basically goes pop, 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 right, 6 times. And those pulses are going to travel, they're sound waves. They travel into the rat. They hit the implant. Power, they provide power. All of a sudden, there's electrical energy running through the transistor, and they reflect back. Okay, so when they come back, the same system that's sending it switches to receive mode. This is all garbage, and captures all of the signals coming back and does a bunch of filtering on them. And what you get is a return. Those 6 pulses come back to us repeated over and over in different packets. And that's because the 6 pulses will first hit one material, then they'll hit the crystal, then they'll keep going and hit another material. And in each of those cases, they bounce back. So you get these bunch of pulses all one after each other. The key is that the pulses that actually hit the crystal powered up the transistor. And if that transistor was connected to or experiencing changes in extracellular voltage potential, it changed what it was doing and changed how the crystal vibrated. And so the reflections changed. That tiny signal in the reflection changed. And so what you can do is from each of these areas where there was a change, we knew which ones they were that were coming from the crystal. We could integrate and sort of do some calculations on there. And I'll show you in the next slide the data. You could reconstruct the extracellular action potential from those changes. And I'll show you how well in a second. Now, I'll skip all the stuff about characterizing noise floor and alignment. You can ask me if you want afterward. But here's the fun part of it, and then I'm kind of done with this part. Top left is wired recording. So that's what we call the reference. It's gold standard. It's something ridiculous. Like we had a 200 kilohertz sample rate, so we had amazing data. It's just completely insane. You don't need anything like that. And those were compound action potentials in that sciatic nerve at different stimulation intensities. And you can see, the more you stimulate, the bigger the compound action potential. And that was what the system was reconstructing. And these are the errors. Basically, it was reconstructing it beautifully. And you could do all the classic sigmoidal recruitment curve stuff that you do when we're messing with it. So this was our very first demonstration that the idea fundamentally worked really well, even, and this is maybe hard to appreciate, even with something as ridiculously simple as a single transistor, as opposed to putting a chip on it and doing all sorts of digital processing, which we'll get to. OK, so that endeavor took on a life of its own, which I'm going to touch on in a second. But at the same time, we immediately realized that you could use these things to stimulate nerves, right? Because I'm sending power to the chip, so all we have to do is, or to the crystal, so all I have to do is build a system that can harvest that power and then dump it into electrodes whenever you want to do electrical stimulation. And this is what we called StimDust. And again, the chip designer for this was Ricky Muller. She's just really good. And so basically, we have this amazing stimulator. There's only 2 or 3 slides on this. Don't panic. We'll go on to other things. But it's really elegant work. So basically, it's the same paradigm, same thing. We're sending an ultrasound. We're powering up this little thing. But now, what the implant does is it has a full chip. That chip is really pretty cool. It's taking power from the piezo. It's taking commands. So now not only do you send in power, but in those pulses, if you change the amplitude of those things, you actually can send it different commands, like do this, stimulate for this long, at this intensity, and so on. And it can report when it bounces back. It's sending back like acknowledgments, like yeah, I stimulated the nerve at whatever intensity you told me to do it, and so on. And so you have these really tiny stimulators now that are extremely efficient. And I won't belabor this. This is really just to give you kind of a snapshot. But you send in these pulses like I did before, but now they have meaning in the sense how wide, how many after you set up a power up, then you have a break, and the chip expects these things to be of different widths, and depending on what widths these are, you're kind of telling it to how much stimuli you want, how big you want the pulse, and so on. And so it becomes, the world begins to open up in the sense that you can apply all the tools of really sophisticated chip design to make intelligent systems that talk back and forth and have a lot of, and then this is all you can look up the Nature paper, this was BME. You can look up these things and look at how good it is. It's really good. You can stimulate pretty much whatever voltage you want for all the useful durations and so on. And it's just a very, very good stimulator, and it's tiny. It's like absurdly small. And this is more characterization of how this thing works. So now I'll use this slide to make one last point. I have 20 minutes, or maybe a little less, maybe 10 minutes, right? About 10 minutes? OK. One of the challenges in these things is that you want to keep the ultrasound lined up. And so the external systems, I mentioned they're sophisticated. To do this, what happens is the system on the outside is actually many ultrasonic transducers in an array. It's very, very similar to the way nose cone radars work on, say, jet fighters. So what happens is you have a phased array of ultrasound, and you have a pretty sophisticated control system that changes the delays on all those things to electronically focus the ultrasound where the implant is. And so you essentially can keep it locked on like a flashlight. Depending on how the signals come back, you can actually calculate how it's moving, how much you should move. There's quite a lot of engineering that can go into these things. You can add IMUs if you wanted to. If you have a hand-operated device and you have hand jitter, the thing can use your IMU measurement to kind of predict. That's the sort of, these plots are really characterizing how good your beam has to be to kind of not lose power and have it work. In addition, you can go very deep. So for this paper, for example, just because of the vagaries of the transducer design, I think they showed something like 5 centimeters. Yeah, about 5 centimeters. Today, it's pretty routine to do 15 centimeters with no problem. So you can go really, really deep which is extraordinary, and I'll come back to that in about 10 seconds. So deep is important. Okay, so this is where neural dust was in terms of what can you do with it. And at that point, and I'll get to the company later, but at that point I knew this had a lot of applications in peripheral neurotherapy. And so I was sitting around thinking, okay, what is it that's left that's a really, really hard problem? And the really hard problem is this. I'd like to be able to do all this through a skull. So I don't think you've seen this. I'm not sure you've seen it. Okay, this is entertaining. So I want to do this through a skull. That sounds crazy, right? Because your skull is a big piece of bone, and primate skull is a big, thick piece of bone that scatters everything and makes a mess out of any of these signals. And so we wondered, what would we do to have this work? And so here's what I want. I mean, I would like to start thinking about how to put this in the CNS. And it turns out I actually don't care so much about cortex. We can get into why. What I think is really a big deal for this are deep structures. No, I mean it technologically, not that cortex doesn't matter. Cortex is okay.
Hippocampus is much better.
That's why this is. Actually, this is for hippocampus basal ganglia. But I mean, cortex, who cares about cortex? So the point is that it's very hard to get into those deep structures in primate. And I think this is where this solution could be a really big deal. So the question as an engineer I want to know is, how many modes can I talk to? Can I get through the whole skull? And then can I go really deep in the primate brain? And I want to show you the quick results. This is a lot of slides, so I'm only going to do a few of them. And I know I'm being obnoxious. It's just that I wanted to kind of have you taste. Let me skip all this. So how do you communicate with many modes? So the red things are transducers that are connected to my interrogator. They're that phased array thing I'm talking about. And all the little blue dots you can barely see are these hypothetical modes. And I recognize that is cortex, but that's how this thing started. The idea being, how do I talk to these things? So you might say, okay, do what you were just talking about, beamform. That's basically, I have them all shoot their beam at this, each of these things one at a time. That turns out to be a really hard problem through skull because it's very hard to beamform. But the other thing you can do, and this is the big idea, the other thing you do is instead I'm going to have all my red external things just launch a plane wave. Just launch a really nice plane wave, have them bounce off of these things, and each of those reflections has a very particular signature that's going to have an effect very different across different receivers, and you'll see them just sort of appear. This is a single receiver, or actually each receiver, the thing. So the idea we started, and this is David Piek, I mean, this is all really his genius, is asking, hey, if I send in a plane wave, I wait for all these things come back, I have a lot of receivers, and I have, and you should all expect this because we live in 2019, 2020, and if I have machine learning, right, that's the answer to everything. So if I put a lot of machine learning on this, can I learn to pick out these different signatures functionally from all the different modes and disambiguate all of them and just learn the whole alphabet of what these reflections do through a skull? And the cool thing is that the answer to this is amazingly absolutely. So I'm going to skip, other than giving you a brief taste, I'm going to skip this. And don't worry, we're nearing the end of the talk. But let's say that this thing here is an extracellular potential that one of these motes is experiencing, say this one. And I'm going to send in some pulses, and here, and I'm listening for the reflections. The blue curve at the bottom is the reflection. So I'm listening for the reflections coming back. And as a particular neuron near one of these things fires, you'll see that the reflections, whatever this looks like, boom, it suddenly changed. And it changes in a weird way. I can't no longer do what I did before, which is a very clean experiment through soft tissue. Like I know what it's always going to look like. I have no idea. All I know is different motes, when they're experiencing a firing event, are going to have some weird differential, all sorts of crazy. And the goal here, and here's, to illustrate this, here's two of them. So here's two different motes, and here's the reflections of both of them, one after the other, in a single receiver. So you see, when one of them fires, let's go back to, wait till the yellow dots come back. So when that one burps, which will happen first, you get a change there, and then this one's doing nothing, and now that one changed, and this one changed very little. Those signatures, they're like fingerprints in the acoustic signature, conceptually should be learnable, okay? And so what we do, I'm going to skip all of this. I know I'm being tedious, I'm obnoxious. What we do is we collect all of these signatures and construct essentially very large matrices of data which evolve over time. So as I'm going, boom, boom, boom in time, taking more of this data, I'm collecting it, And then I'm looking to see where stereotype changes happen. The same thing happens over and over, right? Spiking is sparse, and so I know that must be a spike. And in addition, this allows us to filter out all the movement, because that happens much slower, and you'll see that kind of thing reflected on all of them. So you can basically do very simple filtering to get rid of, sort of compound effects. And you pull out, you learn a library of characteristic modulation profiles, which then, from the raw trace, you build these matrices, and then you infer where there's a spike for each of the motes that's in there somewhere. And now let me show you the punchline. So this is all the blah, 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 matrices and math, and let's just skip all this. So, I'm gonna skip right to the punchline because I'm running late. So, these are all movies. I'm gonna skip the last one. So this is like 3 motes and 1 transducer, 3 motes and 3 external systems. This is what it looks like for 40. And then I'm gonna show it to you through the skull. So basically what's happening is that each of these plots is a single external receiver in a big array listening for blips going on inside the skull phantom. And as these blips are happening, it's learning what different modulations are coincident and pulling out from that a library that allows it to say when an actual spike event occurs. Okay? And obviously I skipped quite a lot to show you this, but the bottom line, this is all EE, I'll skip this too. The bottom line is you can take a macaque skull, build a phantom out of it, and this is for 1, but now we have them for many. The paper is going to have 16. You can have these things embedded, and this is, I have to emphasize, this is what I'm so astounded. There's this, that array that's sitting up there is sitting fully outside the skull. There's no hole in the skull, and it's listening for all this scattered out stuff. And as it finds these little hints over time, it learns a library of them, and it can disambiguate all the firing things. This is where I think, you can imagine where this is going, right? I mean, you can do it this way. You can have a single recording point in the hippocampus connected to an array with many electrodes, and leave it in there and be able to interrogate it from the outside without having any wires, any cannula. So this is very early sort of work, but I think it's gonna have a lot of impact. Okay, I'm gonna skip all this. This is a pretty. Oh, this is a deep dive. Will you guys tolerate 3 more minutes of something else that's fun? Okay. So this is crazy because all the other stuff I can claim I had a finger on. This I have to claim is really just came out of Mason's head, and it's so much fun. I just want to show it to you. So let me skip all this and just give you this. It's another one of these great ideas that's like so simple. So check this out. So if I send an ultrasound wave into any normal medium like water or your brain tissue, whatever, soft tissue, what is that doing? As that wave is propagating, or if it's a standing wave, in the peaks and the troughs, you have pressure differences, right? That's what it means to be a sound wave. It's a pressure wave. And so you have compaction and you have rarefaction, right? You have changes in pressure between the peak and the trough, right? That's just kind of like high school physics. Well, it turns out that the index of refraction of a medium is a function of the pressure. So if I send an ultrasound wave, or I pattern an ultrasound wave in water or gel or tissue, I create changes in index of refraction that follow what the wave is doing. Where there's a peak, there's higher. When there's a trough, there's lower. If I can sculpt this, which we know how to do, how to pattern ultrasound, what you can create inside the tissue essentially are optical components that are completely virtual. So let's imagine you sculpt a circular standing wave where there's a peak at the red stuff and a trough at the blue stuff, okay? And I make that cylindrical, so it's going down in the tissue. I just made an optical fiber. So what you can do is confine light just like you do in an optical fiber without anything, just pure ultrasound. And so this is what Mason showed. This was just published. And the cool part, so one cool thing is that there's you can play games. It turns out these types of optical fibers that you create are called graded fibers, but you can play games in what fraction of photons that are multiply scattered versus, you can play games in the optics. But the cooler part is that you can create these things deep in tissue and then pipe in light into them from external lenses. And what Mason thinks, he's off, running crazy at CMU doing this. What he thinks is that you can, with sufficiently good patterning, which is well within the state of the art, you can create optical, for example, you can create diffraction ratings, you can create lenses entirely virtually in tissue. So you can pipe light around without any pieces of anything. They're purely made out of the ultrasonic confinement of light. And here, let me show you. I was trying to find. Yeah, that's right. That's right. So you're inducing, you can do them traveling waves if you want, but in these things, you're inducing a standing wave of ultrasound, well below any safety issue. I mean, it's really, but it's enough that you get confinement. And so then, essentially this is, shows you how as you increase the intensity, this is data, as you increase the intensity of the ultrasound, your light pipe forms, and now you have this light piped down along the axis of the fiber. And there's all sorts of fun you could do. You could steer the beam. And so here, what he showed was if you then start snapping between different ultrasound modes, you basically can steer the dot around, because what you're doing is just moving around your virtual fiber. You're just sort of aiming it in different locations. So there's a lot of fun to be had. And I want to leave it there. All right, so I'm not going to do this. There's another thing I'm going to stop. I'm going to kill everybody. But you can make neural dust that that measures pretty much anything you want. So this was unveiled last week. It's the world's smallest oxygen measuring optrode. So it's a piece of neural dust that basically measures dissolved oxygen. You can put it as deep as you want, blah, blah, blah. Let me skip all this. So with apologies for being super obnoxious, I'll leave you with the fact that we realized a few years ago now that this had a lot of potential as a platform technology for therapeutics and diagnostics. We launched a company. It was funded very handsomely. It's been off and running for many years. It's about to grow even more. So it's doing very, very well. And I'm going to have a midlife crisis about whether I want to be an academic or not pretty soon. But with that, I'll stop. And I know I went a little fast. So thanks. I'm just worried we're out of time.